Hi students, in today's lecture we will discuss in detail about plant tissue culture techniques. So whatever may be the findings in our laboratory regarding plant tissue culture, we will discuss in detail what are different techniques, what precautions we have to take while performing plant tissue culture, while getting plant tissue culture plantlets in the laboratory. Remember well, as far as plant tissue culture technique is concerned, there may be one question in your mind, what is plant tissue culture? So remember well, it is in vitro culture or in vitro propagation or it is micropropagation. So as far as definition is concerned, the culture of plant seeds, organs, explants, tissues, cells or protoplast on nutrient medium under sterile condition is called as plant tissue culture. What are different bases for plant tissue culture? So remember well, two hormones affect plant differentiation. Either if amount of cytokinin is more as compared to auxin, shoot development is there. Remember well, the amount of auxin and cytokinin, if it is equal, then it leads to the callus development. So this chart will give us a clear idea. Remember well here, this is the particular shoot leaf strip. If amount of cytokinin, if it is more, then it develop the shoot system. If amount of auxin is more as compared to cytokinin, it develop the root system. So the leaf strip, adventitious shoot, root and callus. How this callus develop? So equal amount if it is there, then it leads to the callus development. So this is a general diagrammatic representation of plant tissue culture technique. So first of all, we will take the portion of leaf which is called as tissue or the explant. When we take this explant into the petri dish under all aseptic condition, then after providing all the nutrients into the agar medium or MS medium as per the requirements, which contain micronutrients as well as macronutrients, so it leads to the plantlets. When these plantlets shifted to the field, then we'll get many plantlets after acclimatization. So what are different factors affecting plant tissue culture? We'll see one by one. The first growth factor which is affecting the plant growth, that is minerals. So these minerals are very important as far as growth and culture is concerned. Second factor, that is growth factors. What are different growth regulators or growth hormones which we are adding into the growth media, that is very important. The third one, that is carbon source. If we are going to change the carbon source, directly it affects the plant tissue culture. So second growth factor, that is environmental factors. So these environmental factors play a key role in plant tissue culture techniques, that is light, temperature and photoperiod. So these three factors affect plant growth and development. In addition to that, the next source, that is explant. So here we have to consider different types of explants. Usually the younger, less differentiated explant and the better tissue culture technique leads for different sources of explant. In addition to that, we have to consider genetical factors also. So here in genetical factors, different species show differences in amenability to tissue culture. In many cases, different genotypes within a species will have variable responses to tissue culture. In addition to that, response to somatic embryogenesis has been transferred between melon cultivars through sexual hybridization. So these are all growth factors. In addition to that, what are different explants? So the summary of this particular growth affecting factors, we have to consider growth media, we have to consider environmental factors, and we have to consider source of different explants. So here what type of explants we are taking and what types of genetics we are going to consider for growth and development of the culture. So in addition to that, we have to take care of choice of different explants. So here, desirable properties of an explant is very important. So here, explant we have to select that should be easily sterilizable, then it should be juvenile and responsive to culture media. Then what type of material we have to cut from the different sources of the plants. So we have to remove either shoot tip or axillary buds or seeds or hypocotyls. Hypocotyl we have to select from the germinated seeds or from the leaves. Now here, media composition is very important. When you make an explant like an axillary bud, you have to remove it from the sources of many chemicals and have to resupply these to the explant to allow them for the growth. So remember well, here this is the shoot tip, this is the shoot tip, and this shoot tip is suffered by the combination of auxins and gibberellins. So shoot tip is responsive to auxins and gibberellins. So as far as leaves are concerned, so these leaves are directly, it shows the response to the sugars and gibberellic acids. As far as roots are concerned, so here water, minerals, mineral salts and cytokinins will give response to the development of the roots. So this is regarding the response of the media. So in media we have to add auxins, gibberellic acids, sugars, gibberellic acids, and water, minerals, mineral salts and cytokinins. So what are different medium constituents are required for the preparation of medium? So in the medium we have to add inorganic salts, then we have to add sources of carbohydrates, we have to add vitamins, we have to add water, we have to consider plant hormones. In plant hormones, especially auxins and cytokinins in addition to gibberellic acids. Then solidifying agents like agar medium we have to consider for the solidifying agent. Then in addition to that, there are certain undefined supplements. There are certain undefined supplements we have to consider while preparing the medium. In addition to that, the first component of the medium, that is carbohydrate. What type of carbohydrate we have to consider, how much carbohydrate we have to take. So plants in culture normally cannot meet their needs for fixed carbon, usually added at a source of 2 to 3% of weight by volume. So glucose or a mixture of glucose and fructose is occasionally used. For large scale culture, cheaper source of sugars, especially corn syrup we can take or we may use for the source of carbohydrate. The second, that is photoautotropic culture. So here growth without a carbon source, therefore need to boost photosynthesis. High light intensity is needed for the normal growth and development of a plant. Usually increase carbon dioxide compared to the normal light. So much reduced level of contamination and plants are easily to transfer the greenhouses. So the next component, that is inorganic salts. So what type of inorganic salt we require for the preparation of medium? So it contains a wide range of macro elements and micro elements. A wide range of media are readily available as a spray dried powder. Muracid and Skog medium, so this medium is launched in the year 1965, is the most popular for shoot culture. So in addition to that, Gumberg's B5 medium is widely used for the cell suspension culture. So this leads for no ammonium. So in addition to that, there are certain requirement of vitamins. A wide range of vitamins are available and may be used for the preparation of medium. Generally, the smaller explant, the more exacting the vitamin requirement. The vitamin cocktail is often used, which contain nicotinic acid, glycine, thymine and pyridoxine. So inositol is also used as a supplement for higher concentration of vitamins. So here we'll discuss now what are different plant hormones or plant growth regulators required for the tissue culture techniques. The first one, that is auxins, second cytokinins, then gibberellic acid, ethylene, abscisic acid, and plant growth regulator like compounds we require for the plant tissue culture techniques. The first one we'll discuss in detail, that is auxin. So absolutely essential component. No mutants known from this, and that's why this is very important. Only one compound, that is indole 3 acetic acid, many synthetic analogues, that is naphthol acetic acid, indole butyric acid, 2,4-D, 2,4,5-T. In addition to that, so many compounds we required. Generally growth stimulators which promote the rooting we can add. Then produced in meristem, auxin produced in meristem, especially shoot meristem, and transported through the plant in spatial cells in vascular bundles. The next one, that is cytokinins. So absolutely cytokinin is very essential. So why? Because no mutants known. So single natural compound, that is zeatin, synthetic analogues we can use, that is benzyl adenine, kinetin we can add. In addition to that, stimulate cell division. Cytokinin stimulate cell division with auxins which promote formation of adventitious roots. Cytokinin produced in the root meristem and transported throughout the plant as a zeatin riboside in the phloem. In addition to that, what is the requirement of gibberellic acid or gibberellins? So a family of over 70 related compounds which contains gibberellic acids or gibberellins. All forms of gibberellic acids are required as and when required. So commercially gibberellic acid 3 and gibberellic acid 4 plus 9 which are available in the market for large scale utilization for plant tissue culture technology. So which stimulate the etiolation of the stem. It helps to break down the bud and seed dormancy, and this produce in young leaves. In addition to that, we require abscisic acid also. So what is the role of this abscisic acid? This abscisic acid is only one natural compound which promote the leaf abscission and seed dormancy. So abscisic acid plays a dominant role in closing stomata in response to the water stress, has an important role in the embryogenesis in preparing embryos for desiccation. It helps to ensure normal embryos. So plant growth regulator like substances also required, like polyamines, then jasmonic acid, then salicylic acid. In addition to that, these compounds not universally acclaimed as a plant hormones since they are usually needed at high concentration. So undefined supplements also required. What are undefined supplements? Sources of hormones, vitamins, polyamines like coconut water we can use, or sweet corn extracts also we can use. They are not reproducible and do work very effectively. In addition to that, what are fundamental abilities of the plants? Remember well, Haberland, first time he mentioned that the concept of totipotency, the potential or inherent capacity of the plant cell to develop into an entire plant if suitable stimulated. So it implies that all the information necessary for growth and reproduction of the organism is contained in the cells. So in addition to that, there is one more concept we have to consider for the plant development, that is a differentiation. So plant cell, they are having capacity of mature cells to return to the meristematic condition and development of a new growing point, followed by redifferentiation, which is the ability to recognize into new organs. And in addition to that, the last factor which is available in the plant cell, that is competency. So plant cell is endogenous potential of a given cell or tissue to develop into the particular way. So what are different types of in vitro culture? So culture of the plant that is intact plants, seed orchid culture, embryo culture, organ culture. So this organ culture again are of different types. For example, shoot tip culture, root tip culture, leaf culture, then anther culture. And in addition to that, callus culture, cell suspension and single cell culture, and protoplast culture. Now we'll see one by one. In addition to that, what are different applications of plant tissue culture? So this plant tissue culture technique is very important for micropropagation. Then we can get germplasm preservation by using tissue culture technology. Then somaclonal variations also we can get by the tissue culture techniques. Then embryo culture, we can get haploid or diploid productions. Then in addition to that, in vitro hybridization, for example, protoplast fusion is very important. We can get help, we can take help of plant hybridization. So in addition to that, plant genetic engineering. So what is meant by micropropagation? So remember well, in micropropagation, three important aspects we have to consider. The first one, that is embryogenesis, second organogenesis, and third one, that is microcutting. As far as embryogenesis is concerned, so this embryogenesis we can go for two different methods, direct embryogenesis and indirect embryogenesis. As far as organogenesis is concerned, organogenesis via callus formation, direct adventitious organ formations, then we can get with the help of microcuttings. So what different materials we have to consider for microcuttings? Remember well, meristem and shoot tip culture. In addition to that, bird culture also very important. So this is a very good material cultivated in our laboratory, that is the result of somatic embryogenesis. So in somatic embryogenesis, the process of initiation and development of embryos or embryo-like structure from somatic cells. The production of embryos from somatic or non-germ cells usually involves a callus intermediate stage which can result in variation among seedlings. Not a common micropropagation technique, but it is currently being used to produce superior pine seedlings. So this is the very clear representation of the somatic embryogenesis. So here the development, auxin leads to high production, auxin leads to the high production, then development and cycling of proembryonic masses. What will happen? This is development of proembryonic masses, as a result of which a single cell culture, we have to consider here single cell, then again as a result of which this will lead to the increase of their size and we can get the clear product. We can get the clear product, that is embryo. So what happened from this, when we remove the auxin content, when we decrease the auxin content, that is polyamine interconvergence, so which leads to the development of somatic embryos, and this leads to the somatic embryogenesis. So nowadays the commercial products which are available in the market, that is carrot, then different types of monocots and some conifers we can develop with the help of proembryonic masses as a result of somatic embryogenesis. In addition to that, cleavage polyembryony conifers. So here embryo suspension, embryo, here this is the embryo. So when this embryo we are taking into consideration for suspensor, so it cleave. So cleavage takes place and it leads to the new embryos. It leads to the new embryos as a result of lateral division, So here normal embryo, then we'll get lateral division, and as a result of which we'll get new embryos. So this is the result of cleavage polyembryonic conifers. In addition to this, so here in embryo fermentations, we have to consider somatic embryos may be produced profusely from leaves or zygotic embryos. For micropropagation, potentially phenomenally productive. So shear sensitivity is a problem. Maturation in the liquid is also creates certain problems. So somatic embryos will generate by considering tissue culture techniques. So this tissue culture maintains the genetics of the cell or tissue used as an explant. So tissue culture conditions can be modified to cause to somatic cells to reprogram into a bipolar structure. These bipolar structures behave like a true embryo called somatic embryos. So this is the result of organogenesis. We have produced organogenesis in the laboratory. This is the organs, okay, only shoot development after the combination of auxin versus cytokinin. So in addition to that, what is meant by organogenesis? The process of initiation and development of a structure that shows natural organ form different forms and different functions. The ability of non-meristematic plant tissue to form various organs under aseptic condition, that is called de novo. The production of roots, shoots or leaves is possible as a result of organogenesis. These organs may arise out of the pre-existing meristems or out of differentiated cells. So this, like embryogenesis, may involve a callus intermediate but often occurs without callus. So plant organogenesis, there are two different methods. The first method is indirect method and second, that is direct method. So in indirect method, so this is the pathway include callus stage. So here we have to focus on the callus, the undifferentiated tissue that develops on or around an injured or cut plant surface or in tissue culture. So in case of indirect method of organogenesis, so it bypasses a callus stage and will get the entire organ. So in addition to that, adventitious shoot formation is the de novo development of the shoot from the cell cultures in the absence of pre-existing meristems. So in certain species, many shoots can be induced with this method. So in other species like coffee, it may be necessary to induce an unorganized mass which leads to the formation of callus. So in the somatic embryogenesis and organogenesis, when we compare this, both of these techniques can be used as methods of micropropagation, not always desirable because they may not always result in population of identical plants. The most beneficial use of somatic embryogenesis and organogenesis is the production of whole plant from a single cell, So the new concept, that is microcutting. So this is a specialized form of organogenesis. It involves the production of shoots from pre-existing meristems only. It requires breaking of apical dominance. So microcutting can be one of the three types like nodal, shoot cultures and clump divisions. So as far as micropropagation is concerned, what is meant by micropropagation? The art and science of plant multiplication in vitro is called as micropropagation, usually derived from meristems or vegetative buds without a callus stage, tends to reduce or eliminate somaclonal variation resulting in true clones, can be derived from other explant or callus, but these are often problematic. So all these are the different steps of micropropagation. The first step is very important, that is selection and preparation of the mother plant. So here sterilization of the plant tissue takes place. In addition to that, initiation of culture is very important. Explant placed into the growth medium. In addition to that, multiplication stage is there. Explant transfer to the shoot media and shoots can be constantly divided and it is a divide and redivides. So rooting is there. So explant transferred to root medium and the last stage transfer to the soil. So explant can be returned to the soil and hardened off. That is called as acclimatization. So what are different features of micropropagation? Clonal reproduction, way of maintaining heterozygosity. So multiplication stage can be recycled many times to produce an unlimited number of clones. So routinely used commercially for many ornamental species, some vegetatively propagated crops. Next one, that is easy to manipulate production cycle, so not limited by the field seasons, environmental influences. And lastly, that is disease-free plants can be produced by using micropropagation. So these are key points of this tissue culture application. So first one, that is it helps to micropropagation. Then second, that is germplasm preservation. Then somaclonal variation. Then embryo culture, haploid and diploid production, in vitro hybridization, then protoplast fusion, industrial products from cell cultures, and we can use these plant tissue culture techniques in plant genetic engineering. So next concept, that is germplasm preservation. So this germplasm preservation is very important nowadays for extension of micropropagation techniques. So two methods normally which we are following. The first one, that is slow growth techniques, and second, that is cryopreservation. So in slow growth technique, the proportion of temperature is very important. In addition to that, proportion of light we have to consider. Then media supplements, then tissue dehydration is very important. So medium term storage, that is for 1 year to 4 years. As far as cryopreservation is concerned, so this cryopreservation is the technique used for ultra low temperature, stop cell division and metabolic processes, and very long term or indefinite storage of the cultures we can use. In addition to that, this cryopreservation techniques, what are different requirements? So pre-culturing is very important here. So we have to think about pre-culturing. What happens exactly in pre-culturing? Usually a rapid growth rate to create cells with small vacuoles and low water content. Then cryoprotection. So here cryoprotection can be done with the help of glycerol, DMSO, PEG to protect against ice damage and after the form of the ice crystals. So we have to avoid this ice crystal formation. Next concept, that is we have to consider freezing. The most critical phase, one of the two methods while considering freezing. So slow freezing allows for dehydration, and quick freezing also result in fast intercellular freezing. This is very important. Storage is concerned, cryopreservation storage is concerned, that is with minus 196 degrees centigrade. Then thawing is there, usually rapid thawing to avoid damage from the ice crystal growth. Then recovery we can done. So thawed cells must be washed. Thawed cells must be washed of cryoprotectants and nurse back to the normal growth, which avoid callus production to maintain genetic stability. So the next one, that is somaclonal variation. So a general phenomenon of all the plant regeneration system that involves a callus phase. So two general types of somaclonal variations we have to consider here. So heritable genetic change alter the DNA, wherein stable but non-heritable changes alter gene expression, epigenetic. So here somaclonal breeding producers usually plant cultures as starting material. So idea to target cell in multicellular cultures, usually suspension culture, but callus culture can work. So optional, apply physical or chemical mutagen, apply selection pressure to the culture. So when we are going to target a specific culture, very high kill rate, you want very few cells to survive. So long as selection is effective. So regenerate whole plants by surviving cells. So these are the certain requirements. So effective screening producers. So here in effective screening producers, most mutations are deleterious. then most mutants are recessive, then very large populations are required to identify desired mutations. So as far as clear objectives are concerned, so this particular somaclonal breeding can't expect to just plant things out to see what happens, relates to having an effective screen. This may be why so many early experiments failed. So this is the somaclonal variation. So here see, with the application of different techniques, here change in color of this particular flower. So this is the result. So nowadays we are generating different varieties by changing the morphological characters with the help of somaclonal variation. So this is the parental variety. So in parental variety, all these flowers are white in color, but wherein the ray florets here changes to yellow color. So this is the newly added variety with the production of somaclonal variation. So in plant tissue culture technique, embryo culture is very important. So in embryo culture developed from the, or this embryo culture developed from the need to rescue embryo, the damaged embryo. We can cure this damaged embryo with the help of embryo culture. So these techniques have been further developed for the production of plants from embryos developed by non-sexual methods. In addition to that, this embryo rescue in F1 hybrid form a wide cross, overcome seed dormancy. To overcome immaturity in the seed, to spread generations in the breeding programs, to rescue a cross or self from the dead decaying plant material. So this is the resultant product of embryo culture. So here we have considered, we have taken embryo in this particular test tube. So after providing nutrient material, it is showing the callus. This embryo developed. Then here development of plantlet, the shoot. We have added shoot promoting hormones and root promoting hormones also. And here this plant is ready. This plant is ready to shift into the soil. At last the result of embryo culture. This is the result of embryo culture. Now our plant is ready for transfer into the soil. So this is regarding the embryo culture. So we can produce haploid plants also by embryo rescue of interspecific crosses, then anther culture, then ovule culture also. Like embryo culture, we can culture different embryos, different anthers, different microspores, different ovules. We can get the entire plants. So this is one more method, balsam method. So here the 2n number, here the chromosome number 2n number, it leads to the 14 number of chromosomes which cross with the another variety, and we are here getting the embryo rescue, we have haploid production. Okay, here chromosome number 7. So from 14 chromosome number we are getting here haploid. So haploid production is possible with the help of this technique. So this is very clear anther culture. So this is the entire plant. We have taken here for anther culture, we have taken here one plant. Okay, so from this plant, from this flower, we have removed the anther. So this is the anther. This is the anther. Okay, so from this anther, when this anther we have added into the culture medium, this is the culture medium, culture plate. So in this culture plate, so we have observed here embryoids, different embryoids. So here haploid plantlets. Here we can get haploid plantlets, or the parallel from this anther we will get culture, that is called as callus culture or callus development, and from this callus also we will get new haploid plant. This is called as anther culture. So this is called as anther culture. So in addition to that, from this anther culture, what are different culture factors responsible? So first, that is we have to consider genotype, growth of mother plants, then correct stage of pollen development, then pre-treatment of anthers, then lastly we have to consider culture medium. So ovule culture for haploid production, production is very essential. So this is essentially the same as like embryo culture. So we can develop the melons or onions by following the same growth culture medium. We have to take same growth culture medium and we can develop the melons and different varieties. So what do you think with the haploids? So these haploids, they are weak or sterile plant, usually want to double the chromosomes, creating a dihaploid plants with normal growth and fertility. So we can see the chromosomes can be doubled with the help of these techniques. So all these are the specific examples of plant tissue culture technique by following the F2 generation, and different generations we can produce different types of very economically important cash crops. So all these are the plant tissue culture applications used. So here in today's lecture we have considered applications of plant tissue culture, what is meant by plant tissue culture, how culture medium is developed, then what type of micropropagation techniques, then how germplasm preservation is performed, then somaclonal variations, what are different embryo culture techniques, then haploid and diploid productions, then what is the use of haploid and diploid productions, in vitro hybridization, that is protoplast fusion, industrial products from the cell culture, and lastly this plant tissue culture techniques lead for the production of plants genetically engineered crops. So with this I am going to stop here. So thank you so much.